Hello everyone, and welcome to episode 31, Remastered. To begin this series on the diversity of life, I'm going to start with the oldest, smallest life forms that exist, the archaea and the bacteria. These microscopic organisms cover every living thing, every plant, every animal, both inside and outside. You have bacteria and archaea on your skin, in your mouth, and in your intestines. They exist deep in the soil, they live deep in swamps and in the ocean, and even in extreme environments like hydrothermal vents and super salty lakes. Archaea and bacteria cover the planet, and they're so fundamental that no complex life could readily exist without them. If you listen to the series on the evolutionary history of life on Earth, you should remember that in the first episode, I introduced the Precambrian era. This time period lasted for nearly 4 billion years, representing the vast majority of geologic and evolutionary time. This episode will be kind of like that, in the sense that bacteria and archaea represent the vast majority of life on Earth. Of all the world's domains and orders and classes and families, a majority of them are bacteria and archaea. Of all of the individual organisms on the Earth, the overwhelming majority are bacteria and archaea. Of all life that's ever existed, bacteria and archaea are the oldest, the most fundamental, and the most widespread. It's because bacteria and archaea are so important, so universal, and ever-present in every ecosystem on the planet that I really have no choice but to start this series with them. Life is split into 3 domains. You have the bacteria, the archaea, and the eukaryotes. The eukaryotes are organisms with internal organelles, and many of them are multicellular. Every single animal and plant and fungus are all eukaryotes, as are all of the protists, but I'll cover all of these in subsequent episodes. Today is all about the bacteria and archaea. With that said, let me start with the basics, with how these 2 domains are similar. All bacteria and all archaea are both microscopically small unicellular organisms, tens to hundreds of times smaller than a typical animal cell or eukaryotic cell. Bacteria and archaea are both prokaryotic, which means they don't keep their DNA inside of a nucleus. Where eukaryotic cells have a membrane-bound nucleus in the heart of their cytoplasm, and this encloses and protects their DNA, the prokaryotic cells, like archaea and bacteria, have nothing. Their DNA molecules form circular loops, and they float free and unprotected within their tiny bodies, where they're more accessible to regulatory proteins, but also more vulnerable to mutating factors like UV radiation. Bacteria and archaea both reproduce asexually. Because they live lives measured in minutes or days, they can reproduce dozens of times in a 24-hour period. This means they can quickly produce trillions of descendants, each one a clone of its parent, but with species emerging through an accumulation of mutations over thousands of generations. Even these microscopic organisms can be affected by DNA mutations and the rigorous pressures of natural selection. In this way, bacteria and archaea are an absurdly diverse domain of life. There's around 30 recognized bacterial phyla, but more than 50 if candidate phyla are taken into account. Estimates as to the total range of the bacterial domain reach close to 1,000 phyla, although this estimate is based on the fact that new species are discovered all the time, and so there's the assumption that we don't know how much there is. The archaea can be divided into 4 phyla, although there are also candidate phyla here as well. 2 of the archaea phyla are really extensive, with huge extending classes and families with dozens and dozens and dozens of species within it. But the other 2 phyla are much less researched, and much less is known about them or the lineages within them. If you were to go exploring out into the field and find any organism, be it some species of grass, or a tree, or an insect, or a rabbit, or literally any kind of large, complex life, and you hypothetically took a tissue sample or a blood sample or something and you studied it under the microscope, in every organism, if you extensively analyzed it, you would probably find a new kind of bacterial species, not to mention the dozens or hundreds of known bacterial species that you would also identify. Bacteria and archaea literally cover everything, inside and out. Most of these species of bacteria or archaea that you would find would only exist on those particular plants or those particular animals. For example, your mouth has over 700 known species of bacteria and archaea, and your intestines have over 1,000. After 4 billion years of evolution, the perpetual divergence and speciation and adaptation of all of these bacteria and archaea has created uncountable species, shaped by evolution for an uncountable number of habitats, be it in the soil, on the bark of a tree, on your skin, in your mouth, in your intestines, or anywhere else. And the thing is, if you were to take any of these bacteria species and put them in any other random habitat, like take the bacteria from your mouth and put it on the bark of a tree, it would die, because all of these species are so specialized to their particular habitat. Of those 1,000 or so bacterial species that live in your intestines, a lot of them can only live in the human intestines and nowhere else. So not only will your tissue sample have thousands of bacterial and archaeal species, there's also going to be millions, if not billions, of individual cells. Because these cells are so small, places like your intestines hold many times more bacterial cells than there are your own cells in your own body. If you go outside and grab a handful of dirt, you'll be holding millions of cells of bacteria and archaea right there in your hands. Go to the ocean and scoop up some seawater, and you'll be holding millions of cells of archaea right there in your hands. Huge masses of bacteria and archaea exist deep into the floors of the ocean and deep into the soil of the terrestrial Earth. They exist in hydrothermal vents. They exist in super salty lakes and in hot springs, and they exist across the rugged floor of the world's forests and jungles, and, as I said, in all of the living things of the world. The bacteria and archaea share several major similarities, including their extremely small size, their cell walls, their shared lack of organelles and a nucleus, their ubiquity in virtually every environment, and the fundamentally important and critical role that they play in their local ecologies, be it an animal's gut or as some kind of plant decomposer. But aside from this, there really aren't too many more similarities. When you zoom in to the molecular level, the differences between bacteria and archaea become very apparent. For example, they possess different kinds of biomolecules in their cell walls and their plasma membranes, and they use different kinds of proteins to replicate and express their DNA. I'll describe a few more of these similarities and their differences in greater detail. When I said that bacteria and archaea were small, I don't think small is a strong enough word to describe it. These microorganisms vary in size among themselves, of course, but all of them are absurdly small, as small to your own cells as a wart or a pebble is to you. Bacteria and archaea are measured in micrometers, which are units 1/1,000,000th the size of a meter, or more than 25,000 times smaller than an inch. The smallest bacteria are less than half a micrometer in diameter, which is less than 2/1,000,000ths of a meter. And the smallest archaea are 1/10 of a micrometer, which is 5 times smaller. But in contrast, the largest of them can all be up to 100 micrometers wide. The shapes that they take vary as much as their size. Some shapes are really common, while other shapes are really rare and found only in a small handful of species. Some of the most common shapes are spheres and rods and even spirals, but they can also be shaped like strings, like cubes and tetrahedrons, or like weird globular kidney beans, or flat plates, or other kinds of irregular objects. There's even some archaea that lack a cell wall entirely, and they're shaped like amoeba, like a tomato that was splattered against a wall. Most species tend to exist as independent single cells, but others form distinctive groups or superstructures. The most common arrangements are simple pairings or groupings of a few cells. As far as shape is concerned, the spherical bacteria are called cocci. In pairs, they're called diplococci, and in chains, they're called streptococci. If you've ever had strep throat, you've been infected by a Streptococcus pyogenes bacteria, which under the microscope looks like a bunch of small spherical dots that are all linked together in a squirming line. These little bacteria will infect your throat and your tonsils in their millions, spreading toxins that aggravate and inflame soft tissues. Coughing and even speaking carries air across the inflamed tissue, and it causes the pain that people with strep throat are all too familiar with. Other cocci bacteria, like Staphylococcus, or staph, form random and irregular clusters that are described as looking like a bunch of grapes. Species of Archaea can take even stranger forms, linking together into long filaments or even giant masses. Despite their often similar shapes, bacterial and archaeal cell walls and plasma membranes have very different chemical compositions. Bacteria possess a chemical in their cell walls called peptidoglycan. Those bacteria with a high concentration of peptidoglycan are called gram-positive, while gram-negative bacteria tend to have just a very thin layer of peptidoglycan hidden beneath an external membrane. The classification is a reference to how the bacteria appear under a microscope, after they've been soaked in a purple dye called a gram stain. The gram stain sticks to the peptidoglycan, which makes gram-positive bacteria with their peptidoglycan-heavy cell walls appear dark purple. The gram-negative bacteria don't have that much peptidoglycan in their chemical coating, and so they don't really stain that darkly, if they even stain at all. The gram stain test is really useful for medicine and healthcare, because if we know what the cell wall and the membrane are like, we can figure out ways to disrupt it or penetrate it to kill the bacteria and cure the infection. The Archaea don't have any peptidoglycan in their cell walls. Well, 99% of them don't. There's a group of methanogenic Archaea who have peptidoglycan in their cell walls, but the peptidoglycan molecules they use are heavily modified and quite dissimilar from the peptidoglycan molecules found in bacteria. These archaeal molecules are called pseudopeptidoglycan, and they possess a different sugar, a different kind of linkage connecting those sugars together, and some of the molecules have a different chirality. Chirality is something I think I touched on in the first series, although I might have skimmed over it because it's kind of an intermediate level chemical concept. In either case, the chirality of a molecule describes its orientation. Complex molecules like proteins have very irregular and asymmetrical shapes, but they can exist in two orientations, each one being a mirror image of the other. These aren't perfectly identical copies, they're mirror images. Much like how your face in a photograph is an identical copy, but your face in a mirror is a mirror image. But perhaps a better metaphor is your left and right hands, or your left and right feet. But instead of left and right, the orientations of a chiral molecule are called L and D. In bacteria, their peptidoglycan has D amino acids, but Archaea have L amino acids. The overwhelming majority of amino acids in all living things have the L orientation. Amino acids with the D orientation are much more rare, and they exist really almost exclusively in bacterial cell walls. Chirality isn't the only thing differentiating the biomolecules inside Archaea and bacteria. The molecules they use for respiration, and for metabolism, and for eating and digesting food, huge portions of these molecules are entirely different between the two domains. The internal biochemistry is really where these differences between Archaea and bacteria become screamingly obvious. But before I dive into these smallest details, I should start with something more general, some of the more general differences, and then work my way down the chain of specificity. So, most generally, bacteria and Archaea can be organized based on how they produce ATP for energy. You have Photoautotrophs that make energy through the absorption of light. They're photosynthetic. Then you have Chemo-Organotrophs, which make energy through the oxidation of energy-rich organic chemicals. And then you have the Chemolithotrophs, which make energy through the oxidation of energy-rich inorganic chemicals. Furthermore, bacteria and Archaea consume the carbon that they need to build biomolecules and to replicate through two processes. Autotrophs get their carbon by breaking down simple molecules in the atmosphere like carbon dioxide or methane, while heterotrophs get their carbon by decomposing the organic molecules that are produced by other organisms. These distinctions are important to understand, because every species alive needs to produce energy and gather carbon, not just bacteria and archaea. Because there are 3 ways to produce energy and 2 ways to get carbon, this means that there are 6 combinations that life can use. All eukaryotes, from plants to animals, only use 2 of these combinations. You have photosynthetic organisms like plants and algae, which are photoautotrophs, getting their energy from sunlight and their carbon from CO2 in the atmosphere, and then you have animals, which are chemo-organo-heterotrophs, getting their energy and carbon by digesting the tissue of other organisms. In contrast, bacteria and archaea can utilize all 6 combinations of energy production and carbon acquisition. Because bacteria and archaea were the first life forms, they evolved these first biochemical pathways, the first iterations of photosynthesis, and cellular respiration, and fermentation. With the billions of years of evolution that followed, these processes were shaped by mutations and natural selection, creating a wide diversity of species able to adapt to an equally wide diversity of habitats and ecological niches. And it's interesting to see how 2 of these combinations were particularly successful and led to the emergence of eukaryotes and all of this macroscopic life that we see around us and that we are. These 6 combinations of energy production and carbon acquisition are the photoautotrophs and the photoheterotrophs, the chemo-organo-autotrophs and the chemo-organo-heterotrophs, and the chemolitho-autotrophs and the chemolitho-heterotrophs. Alright, that was a lot of really technical word salad that I just kind of threw at you. Don't worry, it's really not as complicated as it sounds. It's pretty straightforward. Let's start simple with the photoautotrophs. The prefix photo means that they produce energy through absorbing light, or sunlight. All of the phototrophs, therefore, live in habitats where they're exposed to the sun. Those bacteria and archaea that live in the lakes and oceans exist in the photic zone, which is the uppermost layers of the water body, where sunlight can still penetrate deep enough to support life, or I should say to support phototrophic life. The photoautotrophs are those sunlight-dependent prokaryotes that gather their carbon from the CO2 and the methane of the atmosphere, or from whatever substrate they live in. Cyanobacteria are a famous example of a photoautotroph. They live in lakes and oceans and soils, and they use sunlight to power their oxygenetic photosynthesis, and they reduce carbon dioxide for the carbon necessary to grow and replicate themselves. Cyanobacteria exhale molecular oxygen as a result of this CO2 reduction and photosynthesis, and the endless trillions of individual cyanobacteria across the planet that are breathing out oxygen over billions of years has led to an oxygenation of the atmosphere. The cyanobacteria produce the vast bulk of the oxygen that you and I are breathing right now. The photoheterotrophs, on the other hand, cannot reduce carbon dioxide. Instead, they're dependent on consuming organic materials for their carbon. This means anything from the absorption of carbohydrates and fatty acids in soils and sands to feasting on the decaying tissues of dead or diseased organisms. Next up are the chemo-organotrophs and the chemolithotrophs. Both of these groups produce their energy not from sunlight, but through the extraction of electrons from organic and inorganic matter, respectively. The cells take high-energy electrons from their donor molecules and shuffle them from enzyme to enzyme as energy is slowly and safely extracted through chemiosmosis to produce ATP. At the end of this transport chain, the electron has been sufficiently reduced to a low energy state and transplanted onto an acceptor molecule. In eukaryotes, this process takes an electron from an energy-rich molecule like glucose. It produces water as a byproduct, and the electron is finally given to an oxygen acceptor atom at the end of the chain. Many species of bacteria and archaea called organotrophs use glucose and oxygen just like eukaryotes do. But other groups of bacteria and archaea, unsurprisingly, use different molecules and atoms for this process. The methanogens take electrons from molecular hydrogen and give them to carbon dioxide, which produces methane as a byproduct. The sulfur bacteria take their electrons from hydrogen sulfide, often pulling an entire hydrogen atom along with the electron, and they put it on molecular oxygen to create water molecules. The iron-reducing bacteria take electrons from organic molecules and dump them onto iron atoms, literally reducing the iron with the added electrons. The nitrate reducers do the exact same thing, but instead of iron, they dump their electrons onto nitrate, or NO3, to produce nitrogenous byproducts like N2O and molecular nitrogen. There's a tremendous diversity in the chemical foods that bacteria and archaea can use for energy. Because of this huge diversity of chemicals that bacteria and archaea can live on and feed off of, they can exist everywhere. They can consume methane in your gut, or methane from a peat field. They can consume sulfur in a deep-sea vent, or sulfur in volcanic soils. They can persist almost anywhere, altering virtually any habitat with their consumption and respiration. Earlier, I said that cyanobacteria were responsible for producing most of the oxygen in the Earth's atmosphere, but I left out the fact that all kinds of bacteria and archaea are responsible for changing the chemical compositions of the oceans, enriching the world's soils, supporting the biochemistry of more complex organisms, and influencing the fundamental evolution of life itself. When we talk about bacteria and archaea, we're talking about literally the most fundamental species, the most deeply rooted species in all the world's ecologies. One of the most drastic effects that bacteria and archaea had on the world was the oxygenation of the atmosphere. I discussed this briefly in episode 25 on the Precambrian eon, but to summarize, this event was an accelerating catastrophe. As these cyanobacteria produced molecular oxygen, it would fill the atmosphere. At first, this molecular oxygen, which is a very reactive gas, it would have reacted with and bound with iron and other organic materials, other oxygen sinks. But it was only a matter of time until these oxygen sinks were full. When the world's exposed iron and decaying biological material couldn't bind any more oxygen, this excess oxygen began accumulating in the atmosphere. There was nowhere else for it to go. The cyanobacteria kept breathing out oxygen, and it kept accumulating until it got to dangerously high concentrations. I say dangerously because at the time when oxygen was a very rare gas in the atmosphere, oxygen was toxic to most life that was alive at the time, and so the rise of this oxygenic atmosphere killed off most life in an event called the Oxygen Holocaust. Despite the fact that this oxygen killed off a lot of the life that was present at the time, it allowed the surviving life forms to adapt to a world that was saturated with oxygen. It opened the door for life to incorporate oxygen into their biochemistries, to use this electronegative oxygen atom in their electron transport chains to generate chemical energy. Because oxygen has a really high free energy value as an electron acceptor, it allowed newly adapted life to produce significantly more chemical energy than ever before. This abundance of chemical energy would have supported the existence of larger, more complex cells, which would have led to larger, more complex life forms, like the first giant arthropods and the dinosaurs and us. Bacteria and archaea also help to sustain the world's forests and jungles and agriculture through nitrogen fixation. This is because plants use nitrogen as an integral part of their biochemistries. Everything from chlorophyll to their structural tissues to their biomolecules uses a lot of nitrogen, and thus the growth of forest plants and crop plants is often limited by a lack of nitrogen. The plants will grow as much as they can with as much nitrogen as they can access, but when the nitrogen is rare or difficult to access or outright unavailable, growth can slow down or even stop. For example, most of the atmosphere is molecular nitrogen, N2, but this nitrogen is largely inaccessible. The two nitrogen atoms are bound together with a triple bond, which almost no plant or even algae can break apart. They don't have the biochemicals to do it. They don't have the enzymes. Bacteria and archaea are so critically important because they can break apart this triple bond through a process called nitrogen fixation. The lineages of bacteria and archaea that can fix nitrogen have an enzyme called nitrogenase, which is the critical enzyme in breaking N2 into nitrate and ammonia. Unfortunately, nitrogenase can't be exposed to oxygen. If the nitrogenase enzyme comes into contact with oxygen, it gets poisoned and ruined. Because of this, those life forms that have to use nitrogenase are confined to anaerobic environments, or to habitats that are devoid of any oxygen. As a result, these nitrogen-fixing bacteria and archaea often live in shallow water, where they feed bioaccessible nitrogen into the roots of various plants. These kinds of microbes are often used in rice paddies to pump nitrogen into the water, which enriches the plants and bolsters their growth. Nitrogenase-carrying bacteria and archaea also live on dry land, despite being surrounded by the oxygen in the atmosphere. They pull this off by forming symbiotic relationships with plants. The nitrogen fixers, these little bacteria, are encapsulated in nodules along the roots of the plant. Within these small nodules, inside of the plant's tissue, the nitrogenase is kept in an oxygen-free environment, where the bacteria fixes nitrogen that it gives to the plant in return for physical safety. All of this nitrogen-fixing activity has enabled a nitrogen cycle, which is kind of like the water cycle, but it involves plants and animals instead of rain clouds. So in that sense, I guess it's closer to the carbon cycle. The nitrogen cycle begins with the molecular nitrogen in the air, the N2 with a triple bond. This molecular nitrogen gets fixed by bacteria and archaea into nitrogen-containing compounds. These nitrogenous compounds can be decomposed by more bacteria and archaea, and even by some species of fungus, to form ammonia and ammonium ions. The plants can incorporate nitrogen from these sources into their bodies, and the animals that eat the plants can incorporate that nitrogen into their bodies, as do the carnivorous predators who eat those herbivorous animals. When all of these plants and herbivores and predators die, their bodies are decomposed, and the nitrogen is fixed into ammonia. Bacteria and archaea can oxidize the ammonia into nitrite, which can be further oxidized by some bacteria into nitrate. This nitrate can be used in plants, or it can be reduced into molecular nitrogen and put back into the atmosphere, where it starts the cycle all over again. This prokaryote-enabled nitrogen cycle is crucial to life on Earth. Without it, we wouldn't have the forests and the jungles that we have. Plants would struggle to grow, and those that did grow would be stunted. Crop plants would give smaller yields. This nitrogen cycle is clearly the most widespread and important chemical cycle for plant life, but bacteria and archaea are also involved in cycling chemicals like sulfur, phosphorus, and carbon. It's not all sunshine and roses, however, as human interference in the nitrogen cycle through artificial fertilizers and industrial agriculture often leads to nitrogen pollution. This is a very complex problem that affects life at many levels in an ecosystem, so try and follow along with the path of pollution that nitrogen takes. Let's start with an average plot of farmland that gets saturated with nitrogen in the form of fertilizer, which is laid into the soil to fuel the growth of the bacterial and archaeal communities that live there. A lot of the nitrogen in the fertilizer gets converted by these microbes into nitrate, and the nitrate, being soluble in water, is easily flushed out of the soil through rainfall, or just by the farmer watering the farmland. Unfortunately, these nitrates accumulate in rivers and groundwater sources like wells and aquifers, and then they get funneled into the oceans. The nitrates are consumed by bacteria and archaea in the oceans as food, and their populations explode. More food means you can support more people, or more individuals, and this applies to bacteria and archaea just like it applies to humans and everything else. When the swollen bacterial populations reach maturity and die, their microscopic corpses will litter the ocean floor in their trillions. On the ocean floor, even more bacteria and archaea can decompose the bodies of their dead cousins, and they experience a similar but delayed boom in their populations. As these seabed-dwelling bacteria and archaea decompose the organic matter and reproduce on their own, They consume an absurd amount of oxygen, and this is where things get pretty bad. This massive overconsumption of oxygen in these situations can create massive anoxic dead zones in the water, areas that are deprived of oxygen, where fish and other oxygen-dependent organisms can't live. There are literally vast expanses of anoxic water, filled with no life at all, save for any anaerobic microbes that happen to be floating around. These dead zones can be catastrophic for marine ecosystems and the fishing industry in general. It's like habitat loss in the ocean. For example, the American Midwest is covered in extensive farmland, and all of this farmland uses a copious amount of fertilizers, and these fertilizers end up creating nitrates in the soil because of all of this bacterial activity. But these nitrates can get washed away just by irrigation or rainfall or flooding, where they all get washed into the Mississippi River, and as they flow down the river they get deposited into the Gulf of Mexico. And all of these nitrates in the Gulf of Mexico have created an anoxic dead zone the size of New Jersey. The bacteria and archaea in our intestines can also be thrown out of equilibrium by human interference. The gut flora play a huge role in maintaining the health and vitality of a human being or any other animal. The bacteria and archaea in our stomach and intestines help break down food. They help digest our food and make sure as many kinds of biomolecules get broken down as possible, so we have this optimal nutrient yield. They can even help in areas not directly related to digestion, like supporting the immune system and producing hormones for fat storage. When you consume antibiotics or probiotics, you're altering your gut flora. Antibiotics can damage and kill bacteria and archaea, and if you take too much of them or you take them too often, you're not following your prescription's directions, you can destabilize the ecology of your gut flora by wiping out certain species. This can really irritate your bowels, and by killing off the gut flora that boost your immune system, you can make yourself more susceptible to diseases. Because bacteria and archaea are involved in the digestion of carbohydrates, their depletion and removal from the gut can lead to the incomplete digestion of carbohydrates. These incompletely digested carbs can soak up water, which leads to watery stool or diarrhea. Because of these and other related issues, antibiotics have to be consumed either in careful doses or in combination with probiotics to counter the negative effects. Now, I'm almost at the end of the episode, and I haven't even mentioned the diseases directly caused by bacterial or archaeal infections. As virtually everyone in the modern world knows, bacteria and archaea can infect you and cause disease. They do this by penetrating your body's defenses, like your skin and your immune system, and interfering with your cells to compromise your health. This can happen through a huge variety of mechanisms, so many that it would be literally impossible for me to list them all. So, to keep it brief, bacteria can infect other things and harm them by releasing toxins that interfere with the proteins of healthy cells, or that disrupt the membranes of organelles, or that interfere with some kind of regulatory process or chemical gradient or chemical pathway. These toxins and their interfering effects can wound cells and make them unable to function properly, or these toxins can just outright kill an infected cell. The immune response to a bacterial infection causes swelling and inflammation, and this can cause more damage to the innocent cells that are getting involved in the pathogen attack. The immune system can fight back by identifying specific chemical tags on the exterior surface of the bacterial or archaeal cell, and by mounting armed responses to the invaders, they can remove the pathogens. Now, that's almost an offensively simplified explanation of the immune system, so I feel compelled to tell you guys that I will cover the immune system in much more detail in a future episode. With that said, I think that's about all that I have for you today. So to recap, bacteria and archaea are the oldest and most fundamental life on Earth. They cover everything, and they live everywhere. They're involved in almost everything, and just by the nature of them existing and eating and breathing and doing their thing, they literally power the numerous chemical cycles that define life on Earth, from nitrogen cycling to phosphorus and sulfur cycling to carbon cycling. Although they're small, and they often look similar to the untrained eye, bacteria and archaea are vastly different creatures on a chemical level, with different molecules in their membranes and their cell walls, with different means of producing energy and accumulating carbon to grow and reproduce. Bacteria and archaea are integral for your life and mine. They let us digest food and stay healthy and happy. So the next time you eat a meal, try to appreciate the fact that the energy from this food, the energy that will keep you alive and help your body maintain itself, is all largely being processed and delivered to your cells by the hard work of the bacteria and the archaea that line the inside of your gut. I hope this episode was fun and informative, and if you thought so too, give it a like. I hope you learned something cool, and I hope you're enjoying all of my content. And if you are, hit the subscribe button to get new stuff every Monday. And as always, thanks for listening.